Good evening, everybody. Myself, S. Ramachandran, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Vistas, Chennai, is going to present a seminar on the topic lipids. Lipids are class of biological molecules defined by low solubility in water and high solubility in non-polar solvents. They are molecules that are largely hydrocarbon in nature. Lipid represents high reduced forms of carbon and upon oxidation metabolism yield large amounts of energy. Lipids are thus the molecules of choice for the metabolic energy storage. Lipids are mostly small molecules and not polymers, unlike polysaccharides, proteins, and nucleic acid. The classification of lipids is based upon the structure, nothing but the simple lipids, complex lipids, or nothing but the compound lipids, the derived lipids, the miscellaneous lipids, and the natural neutral lipids. The classification of lipids is also based upon the hydrolysis reaction in alkaline solution, saponification lipids and or saponifiable lipids and non-saponifiable lipids. Saponifiable lipids can be hydrolyzed by alkaline condition to yield salts of fatty acid, whereas non-saponifiable lipids does not undergo hydrolysis reaction in alkaline solution. This classification of lipids is based upon the structure, which means energy storage lipids, triacylglycerols, membrane lipids like the phospholipids, emulsification lipids like the bile acids, the messenger lipids like the steroid hormones, and the protective coating lipids like the biological waxes. Biological functions of lipids are most important. The most important role of lipids is it acts as a fuel, or fat is the most concentrated form of energy that can be stored. Since fat is a bad conductor of heat, it provides excellent insulation. Lipids are the constituents of membrane structure and regulate the membrane permeability, like the phospholipids and the cholesterol. Fat may also provide padding to protect the internal organs. Some compounds derived from lipids are important building blocks of biological active materials. Lipoproteins are constituents of cell wall. One more important function of dietary lipids is that it is of supplying so-called essential fatty acids, LAL, linoleic, and arachidonic acid. They are the source of fat-soluble vitamins, nothing but the A, D, E, K. Lipids are important in cellular metabolic regulation, nothing but the steroid and the prostaglandin. These are some biologically important fatty acids, nothing but the saturated fatty acid and the unsaturated fatty acid. Saturated fatty acid contains a single bond, whereas unsaturated fatty acid contains a double bond. It can be a double bond, conjugated double bond, or isolated or non-conjugated also. The physical properties of fatty acids, nothing but the length of the carbon chain in a fatty acid is important in determining things like your water solubility and melting point and the boiling point. Long carbon chains are non-polar and things with long carbon chains on them do not dissolve in water. Short chain fatty acids are slightly soluble in water because of the presence of carboxyl group, which is polar in nature. The triglycerols are nothing but the glycerides, which are lipid esters. It contains the alcohol group of glycerol to form ester with fatty acid. Esterification may occur in 2 or 3 or alcohol position producing monoglyceride, triglyceride, and diglycerides. The triglycerides are insoluble in water, widely distributed in plants. Triglycerides are non-ionic and non-polar. Triglycerides serves as energy storage in adipocytes of adipose tissue. This is the classic example of a glycerol, which is nothing but contains the triacylglycerol, whereas the alcohol is replaced by ester molecule, where R contains the higher fatty acid. Example, a structural example of triglyceride. Chemical properties of triglycerides: hydrolysis, saponification, hydrogenation, rancidity, antioxidant. Hydrolysis is a breakdown of triacylglycerol. Saponification value forms of glycerol and your formation of soap. And hydrogen addition of hydrogen to the molecule to form a saturated fatty acid. Rancidity, deterioration of fats and oils resulting in unpleasant taste. Ultimately, it gets oxidized, and antioxidant that prevent the occurrence of oxidation rancidity. Next is lipid peroxidation. So to find out the purity of fats and oils, you have various measurements like the iodine value, defined as the grams of iodine absorbed in 100 grams of fat or oil. Saponification number, nothing but is the number of milligrams of potassium hydroxide required to completely saponify 1 gram of oil or fat. Richard Missel number, nothing but is the number of milliliters of 0.1 N potassium hydroxide required to neutralize the distillate obtained from saponification, acidification, and steam distillation of the fat, or 5 grams of fat. It is useful in testing the purity of butter as it contains a good concentration of volatile fatty acid, nothing but butyric acid, caproic and caprylic acid. Acid number, number of milligrams of potassium hydroxide required to completely neutralize the free fatty acid present in 1 gram of oil. It is used to find out the presence of double bonds and triple bonds present in the given compound. Phospholipids, they are the compounds or complex lipid containing phosphoric acid, fatty acid, nitrogen base, and alcohol. It contains of many classified as glycerophospholipids, sphingosphospholipids. Phosphoglycerides are further classified into choline, ethanolamine, and serine. Types of phosphoglycerides are phosphatidic acid, lecithin, cephalin, phosphatidylinositol, phosphatidylserine, plasmalogens, and your cardiolipin. And most importantly, the sphingomyelin. Sphingomyelin is amino alcohol present and do not contain glycerol. Sphingomyelin attached to and by amide linkage to a fatty acid to produce ceramide. Next comes the glycolipids, presence of glucose molecule in it. In that you have many types. You have the ganglioside. Sphingolipids storage diseases are Stay-Stack disease, Gaucher's, your Krebs disease, and Niemann-Pick disease. Niemann-Pick disease is most commonly white storage diseases found. Next come to the very important part, which is called as the lipoprotein. Lipoproteins are molecular complexes of lipids with proteins. They transport vehicles of lipids in the circulation. There are 5 different types of lipoproteins: chylomicron, LDL, low density lipoprotein, VLDL, very low density lipoproteins, HDL, high density lipoproteins, and the free fatty acid albumin complexes. Next comes the steroid, very important, contains the cyclopentanoperhydroxypyrazine ring. The common steroid known is nothing but the cholesterol, very important. It is the most abundant steroid in the body, insoluble in water and need water molecule carrier. Has methyl groups, alkyl chain, hydroxy group in the steroid nucleus. The classic structure of cholesterol is present. You can see you have the hydroxy group at the third position, methyl group, 2 methyl groups, and the side chain. Cyclopentanoperhydrophenanthrene ring. You have many other steroids, nothing but your cholesterol compound, nothing but ergosterol, sigmosterol, phytosterol, all this one, aldosterone, corticosteroids, all these comes in steroids. What is the function of cholesterol? Poor conductor of heat, provides insulation. It provides in the functions and the maintenance of regulation of the membrane structure, and it is used in the biosynthesis of bile acids and sex hormones. Last but not the least, you have the waxes. It's a higher molecular weight compound. The monoester formed by the reaction of long chain monohydroxyalcohol with fatty acid molecule. It gives a long chain. It is having high molecular weight. Therefore, it is called waxes. Best example is the beeswax. And the biological law of waxes is protective coating on leaves, stems, and fruit of plants and the skin and the fur of animals. Uses carnauba wax and lanolin are examples of waxes which are used prevalently. These are the references that I use for my seminar. Thank you.